Hello everyone, this is your YouTube channel Family Diverse. Now we are focusing upon the beta oxidation of a fatty acid. Beta oxidation of fatty acid, also known as fatty acid oxidation. What is the definition of beta oxidation? The the oxidation of fatty acid on beta carbon atom. This result in a sequential removal of a two carbon fragments acetyl-CoA. This is known as beta oxidation. Uh, Now, in all the tissues, the fatty acid goes into oxidation. In specific tissues, it does not utilize fatty acid or does not go beta oxidation. In brain cells and erythrocytes, means the RBCs and adrenal medulla cannot utilize fatty acids. So, beta oxidation take place in three steps. First of all, we have to activate activation of fatty acid occurring in the cytosol. Then we have to transport that fatty acid from cytosol or a cytoplasm, from cytosol to into the mitochondrial matrix. Then we have a beta oxidation proper in the mitochondrial matrix. Activation of fatty acid occur in the cytosol. Fatty acids are activated to acetyl-CoA by thiokinase or acetyl-CoA synthesis. This reaction is occurring into two steps in which is required ATP, coenzyme A and magnesium ions. The fatty acid react with ATPs to form acyl-aldolate, then which combine with coenzyme A to produce acetyl-CoA. In this reaction, with the help of thiokinase or acetyl-CoA synthesis, the ATP molecules is utilized. In that, adenosine monophosphate is utilized. Then we have two phosphates, pyrophosphate. Then it goes into phosphatase enzyme to form phosphate. Uh, Note, three different thiokinases to activate long chain 10 to 20 carbon, medium chain 4 to 14 and short chain less than 4 carbon fatty acid have identified. Now we are focusing upon first step, activation of fatty acid occurring in the cytosol. This is our fatty acid. This is COO I minus ion. After that, first carbon, alpha carbon, then beta carbon. With the help of thiokinase enzyme or acyl-CoA synthase enzyme, ATP is utilized here. Here, ATP monophosphate is utilized. Then we have two phosphates. In ATP, adenosine triphosphate. That's why pyrophosphate. Then pyrophosphatase. With the help of pyrophosphatase enzyme, it's formed two phosphate. Then acyldenylate is with the help of coenzyme, it's remove adenosine monophosphate to form acyl-CoA. Then we have our second step of beta oxidation, transport of fatty acid from cytosol into the mitochondrial matrix. The inner mitochondrial membrane is impermeable to the fatty acid, a specific carnitine carrier system which operates to transport activated fatty acid from cytosol to mitochondria. It's occur in four steps. First step, acyl group of acyl-CoA is transferred to carnitine. This is the structure of carnitine, beta hydroxy gamma trimethyl aminobutyrate. It's catalyzed by carnitine in acyl transferase 1 enzyme. This enzyme is present on the outer surface of the inner mitochondrial matrix. And we have second step in the step 2 of beta oxidation. The acyl-carnitine is transported across the membrane to the mitochondrial matter with the help of specific carrier protein. Carrier protein. In the third step, uh, in this step, carnitine acyl transferase 2 is converted into acyl-carnitine to acyl-CoA. This enzyme found on the inner surface of inner mitochondrial matrix. Then the carnitine release returns into the cytosol for reuse. Now we have acetyl-CoA in cytoplasm. We have seen the activation of uh, fatty acid occurring in a cytosol. So we form the acetyl-CoA. From acetyl-CoA, this is acetyl-CoA. It's present carnitine system here. So with the help of carnitine, with the help of enzyme carnitine acyl CoA transferase 1, the CoA will be released, then the carnitine is attached. Here is the structure of acyl carnitine. Then with the help of carrier protein, it transports into inner mitochondrial membrane to mitochondrial matrix. Then with the help of carnitine acyl transferase 2, it will release carnitine and form acyl CoA with the addition of CoA enzyme. Now we have to go for the step 3, beta oxidation proper in the mitochondrial matrix. Each cycle of beta oxidation liberates a 2 carbon unit acetyl CoA occurs in a sequence of 4 reactions. We have oxidation and cleavage. In the oxidation, acyl CoA undergoes dehydrogenation by an FADH dependent flaval enzyme, acyl CoA dehydrogenase. A double bond is formed between alpha and beta carbon. In this step, FADH₂ is formed, which goes into ETC cycle to form 2 ATPase. I want to say one thing about that. In wherever the dehydrogenase enzyme is there, the ATP is utilized or the ATP is generated. You see, acyl-CoA. With the help of acyl-CoA dehydrogenase enzyme, FAD is converted into FADH2. Then it goes to ETC cycle to form 2 ATPs. So here is the removal of one hydrogen from here and one hydrogen from here and one hydrogen from here. This is alpha carbon, this is beta carbon. Then to form trans-enol CoA. Then we have second step in Enyl CoA hydratase brings about hydration of the double bonded to form beta hydroxyacyl CoA. In this step, addition of water molecules by enzyme enyl CoA hydratase, which breaks the bond between alpha and beta carbon. On a beta carbon, OH is attached and hydrogen is attached to the alpha carbon of a beta hydroxyacyl CoA. Now we have trans enyl CoA. With the help of enyl CoA hydratase, one molecule of hydrogen is added. In that we have H plus and OH minus ion. We are doing beta oxidation, that's why the OH molecule is attached to the beta carbon OH. Then alpha, for in alpha carbon it's attached to hydrogen. So that's what the bond is breaked between double bond is converted into single bond. Then we have an oxidation. So it's form beta hydroxyacyl CoA. Now we have oxidation, second oxidation step in a beta oxidation stage. The beta hydroxyacyl CoA undergoes the reaction with beta hydroxyacyl CoA dehydrogenase to form the product beta ketoacyl CoA. This is the second oxidation in which it generates one molecule of NADH plus H plus. Then it goes to ETC cycle to form 3 ATPs. The two hydrogen removed from the beta carbon of beta hydroxyacyl CoA. See here. Now we have beta hydroxyacyl CoA. This is beta hydroxyacyl CoA. From there, with the help of dehydrogenase enzyme, beta hydroxyacyl CoA dehydrogenase NAD plus NADH plus H plus, it goes to ETC cycle to form 3 ATPs. The removal of one H from here, one H from here, so it's converted into keto group beta carbon, so beta keto acyl CoA. Then last and final step is cleavage. The final reaction in the beta oxidation is the liberation of two carbon fragments acetyl-CoA from acyl-CoA. This occurs by the thiolase enzyme. It is a thiolytic cleavage. The enzyme also known as beta keto acyl-CoA thiolase. Here, beta keto acyl-CoA with the help of beta keto acyl-CoA thiolase enzyme, its cleavage occur and formation of acyl-CoA and acetyl-CoA. This acyl-CoA goes to the first step of oxidation, beta oxidation. It goes to this step and form up to less, and its form this acyl-CoA goes to the first step of beta oxidation of proper beta oxidation proper in mitochondrial matrix. That is first oxidation until the number of carbon is less than 4 carbon of fatty acid. Then this acetyl CoA goes to the Krebs cycle and liberates 2 carbon dioxide. Now we will see the net product. Now we will see palmityl CoA is 16 carbon. The net product 7, we have 7 NADH plus 7 FADH₂ and 8 acetyl CoA. 1 NADH plus and H plus form 3 ATPs. 3 into 7 21. FADH₂ 2 into 7 14 ATPs. 8 acetyl CoA form 12 uh, 8 acetyl CoA from 12, so 96 ATPs. Then the energy utilized for the activation is 2, so 129. The total net ATPs are 129. For 1 molecules of palmityl CoA, it generates 129 ATPs. Now stay tuned another updates, upcoming updates, and if you want for the notes for beta oxidation, please check out my Telegram channel. I will provide there. Thanks and subscribe to my channel.